So in the last class we have seen about the different steps of canning. What are the different steps how to make the canned products? So in today's class we are going to see the different spoilage of canned foods. Before getting into that let's understand what are the different categories of canned products. One is the pasteurized product. Pasteurized product means we will not be subjecting to high temperature. Say we will be normally subjecting the product to below 100 degrees centigrade. So we will be just giving a slight thermal treatment. So F0 value, all these things will be dealt in the coming class. F0 values is almost 0. What is F0 value? We'll be talking in the next class. Is it clear? So what we are doing here is we inactivate most of the vegetative microorganisms. So we are not subjecting to high temperature, we will be subjecting to below 100 degrees centigrade only. The core temperature is in the range of 83 to below 100 degrees centigrade. That core temperature of the can, okay, the geometric center, that I hope you remember that critical thermal point. Okay, so here since we are not subjecting to high temperature, we will be destroying only the vegetative microorganisms. And the spores of Bacillus and Clostridium or thermophilic bacterial spores are not destroyed. Okay, because if you want to destroy the spores, we have to subject to high temperature. Then the storage requirement. So in this product, the pasteurized product, it requires uninterrupted cold chain. So we have to maintain the cold chains in the pasteurized product. So what is the shelf life? Up to 6 months. Okay, so what is the advantages you see here is ordinary if you keep the meat in the refrigeration, we can keep it for 4 days or 5 days. But the pasteurized product, we can keep it almost for 6 months. So this will extend the shelf life. Okay. So otherwise, if you want to keep for 6 months means we have to keep it in the freezer. So here we are saving the energy. Just by keeping at 0 to 4 degrees centigrade, we can keep it for 6 months. So what type of products are generally pasteurized? Okay, so here most of the pork product, we can say, especially the ham, the bacon, or generally we go with pasteurization. We don't go with sterilization. Okay, so why? The reason is that when you are subjecting to high temperature the pork products, it will give a metallic taste. That is the reason why generally the pork products are pasteurized and it is not sterilized. Is it clear? So we have to write in the label also, it is perishable and must be refrigerated. So in the labeling we have to give it is a perishable product and must be refrigerated. You cannot keep in the ambient temperature. Generally people think the canned products means we can keep in the ambient temperature. But the pasteurized canned products, you have to keep it in the refrigerations and then only you can keep it for 6 months. So this is pasteurized product. Remember the F0 value is almost 0. What is F0 value? We'll be discussing in the next class. So next is the sterilized product. So here we will give the intensive treatment and the core temperature of the product is approximately, it will attain around 115 to 121 degrees centigrade. Okay, and the F0 value is most important here. The F0 value is 4 to 6. Okay, we will be attaining the F0 value of 4 to 6 in the sterilized product. So what are the things that we inactivate here is that we will be inactivating all the vegetative microorganisms except the thermophilic spores. Okay, all the vegetative microorganisms except the thermophilic spores. Non-thermophilic spore formers and all other things which will be inactivated here. So what we are not inactivating, again the spores of Bacillus and the Clostridium or the thermophilic spores are not inactivated. The storage requirements. So again, you can store this product at a ambient temperature. Okay, we can store this product at a ambient temperature. Shelf life is almost 1 year, but we cannot store above 40 degrees centigrade. The reason is that because if the spores are present, if the Bacillus or Clostridium spores are present, then it will start germinating. So if we keep above 40 degrees centigrade, say normally the most favorable is around 42, 43 degrees centigrade. To be on safer side, we say 40 degrees centigrade. So if you go to Gulf countries and if you are keeping this canned product at ambient temperature, so you know that in the Gulf countries, the temperature of the atmosphere, the surrounding temperature will be around 50 or 46 or 48 degrees centigrade. So if you keep it at that temperature, there's a chance for the spores to germinate, and it can form vegetative cells, and it can multiply, and it can produce the toxins, or it can brings to the spoilage. Is it clear? So that is why it is we advise them to keep below 40 degrees centigrade. Shelf life up to 1 year, but not suitable for tropical countries. Conditions means where that environment temperature is above 40 degrees centigrade. We are not recommending the sterilized products. Then the spores will not, because but not tropical countries, because the spores will not germinate below 43 degrees centigrade. You understood that? So if you keep in the above 40 degrees centigrade, since our F0 value is only 4 to 6, then there's a chance for the spores not get destroyed. So there's a chance for the spores to germinate. Labeling must be refrigerated once it's opened. So any canned products, once it is opened, you have to refrigerate it. Is it clear? Once, suppose a can of 300 grams, okay, we are just taking 100 grams, and again you cannot keep it in the ambient temperature. Again, once it is open means you have to keep it in the refrigeration. So next is commercially sterile product. So we have the pasteurized product, we have seen pasteurized product, sterile product, and commercially sterile product. So what is commercially sterile products? Here we are giving the intensive thermal treatment. So we are subjecting the product to 115 to 121 degrees centigrade, and the F0 value or thermal dead time, okay, it is more than 12 and above. Okay, the F0 value will be more than 12 and above. So such type of product is called as commercially sterile product. So what we are inactivating in this process, we inactivate all the vegetative microorganisms, including all the spores, that is thermophilic spores also we will be destroying. So the storage requirement, ambient temperature, you can keep it. You can even keep it at a tropical conditions above 40 degrees centigrade. You can keep it. So you have the shelf life of around 3 to 4 years. So this is called as commercially sterile products. So the labeling must be refrigerated once it is open. Okay, in the pasteurized product, always you have to keep it under refrigeration. In the sterile and commercially sterile products, we have to once it is open, it has to be kept in the refrigeration. So we can keep it for another 2 or 3 days like. Okay, that is the thing that we have to put it in the label. So I hope it is clear what is pasteurized products, sterile product, and commercially sterile product. So how the normal cans and the hard swell cans. So this is the normal cans. You can see this is the normal cans. It will be always concave. The surface will be concave. Already we discussed about this. So once it is swells, then it is a blown can, or it is a what you called as a unhealthy cans, we can see. Okay, so this is a blown. So normal cans, whenever you purchase the can should be concave, or it should be somewhat straight. Okay, but here it is convex. You can see it is convex. So this is called as a blown can, or the bulged cans, or the swell cans. So again you can see here. So you can see a swelling. So when it swells in the last stage, the seamings will break. The seamings will break. Okay, and sometimes blow off. Okay, that we'll see. So when it will get this type of cans, okay, when we can get this type of cans, we can see one is overfilling. I already told that there should be sufficient edge space, because the product will swell when you are subjecting to high temperature. So we have to give the edge space of 0.6 to 9 centimeter, 0.6 to 0.9 centimeter edge space we have to give. Already we talked about that. If you are doing overfilling, and when you are subjecting to high temperature, what happens is the cans can swell. That is one thing. Another is microbial spoilage. So if it is because of sometimes microbial spoilage, some bacteria may produce the gas. So it is, if you are not processed properly, under processing. Okay, under processing. If you are not subjecting to proper temperature, time, if you are not subjecting that, the microbes may not be destroyed, or the spores may not be destroyed. That is under processing. And when it get a favorable condition, the spores may germinate, or the vegetative cells will multiply, and they can produce the gas. So in that cases also it can bulge. Microbial process from the post-processing contaminations. That is, sometimes what happens is there may be minute holes, like, okay, holes can be formed, or the seamings may not be proper. Okay, or the seam, this is double seamed cans. Sometimes what happens is the seaming may not be proper. What happens is through that small holes, the bacteria can enter. That is post-processing contaminations. That can also leads to this type of bulging. So microbial gas production during the lag phase between the closing and processing. Is it clear? So another problem that we can come across this, we fill the cans, seamings, everything will be done, but it takes more time for retorting. So if you keep the cans for, if you are unhygienic, it is done like, and if you are not going for processing immediately, what happens is there will be some time for the bacteria to produce the gas. During that time also we can come across this type of bulging. Another is hydrogen gas production from a chemical reaction of the product with metal plate. We'll see that one. So always remember that the healthy can will be concave and unhealthy can, or the blown cans or the swelled cans, will be convex. So what is buckling? Buckling means it's a permanent deformities of the can's ends. Okay, can's ends may the seamings may break, or the hence the lid will be breaking. So why it occurs if the retort pressure is released suddenly after the cooking period. So that's what I told. After sterilization, we will be condensing the steam around the cans. Okay, you remember that or not? So when you inject the water, what happens is the surrounding steams will condense faster. So the inner pressure will be higher than the outer pressure. The pressure inside the can will be higher than outside. Is it clear? So if you are not counteracting that pressure, what happens is the cans can break, or the seams can break. That is called as buckling. Okay, that is called as buckling. Then corrosions. Okay, this is what I told you. Sometimes we can come under the corrosions can take place, or the tin coating is not proper. So when we are exposing to more moisture or humidity is high in that environment where we keeping the cans, so that cases it also leads to the corrosions. Okay, corroding will be taking place. You can see here rusting. Okay, you know that when iron is exposed to oxygen and moisture, the rusting will take place. That's why we do painting. Is it clear? In the case of cans, we are coating with the tin. Okay, if the tin coating is not proper, or if there is a small break or small breaking in the tin coating, from there the rusting will start taking place. Is it clear? So that is what we have to take care. So why the wet cans? Due to either excessive post-process cooling. So when you are cooling out, I told you when during the cooling processes, we have to keep between the 36 to 39 degrees centigrade. Why with 36? Because most of the moisture that is condensed on this will be evaporated at 36 degrees centigrade. So if you are doing more cooling, okay, if you do more cooling immediately, what happens is the moisture will be condensing. If the tin coating is not proper, from there the rusting will start. So if you're not cleaning properly, okay, we have to wipe out the moisture and everything. That's what I told during the processing. We have to clean the cans properly, and then we have to go for labeling. If the cleaning is not proper, if the person who is involved in cleaning is not taking much care and immediately go for labeling, happens? So in such conditions, the rusting can take place. Improper temperature. If you are not maintaining proper temperature and humidity, so if there is high humidity in the environment, okay, so if you are storing in such places, what happens is there is a chance for rusting. Cans unprotected from weather during transportation or storage. That's what I told you. Humidity is more, moisture will be more. Naturally, if the tin coating, even a small place, if the tin coating is not proper, from there the rusting will start to begin. Okay, so this is what we have to take care. Double seaming defects, you can see here only the first operation. So here sometimes what happens is when you do the seaming, I already told the cans we go with the double seaming. So if the seaming is not proper, one seaming will be done, next seaming is not taking place properly, then we can get a small holes here, gaps. It's called normally in the cans we follow double seaming. Already I talked about during the processing. So here what happens is the seaming will not be proper. Through that small holes, the air can enter or the microbes can enter. So the seaming is not proper, the moisture will be there, it will start rusting like. Okay, this is called a double seaming defect, double seam defect we can see. So you can see here, okay. So we have to seal it properly. Coating application defects, that's what I told you. During the tin coating, if some dusts are there, okay, if some dust, when they're doing the coating of the tin plates, what happens is if some dust is there, that place it will not be tin coated, will not be there. and from there the coating will not be there, so the rusting can take place. So we have to take care of all this. This is a manufacturing defects in the cans. Okay, this is a manufacturing, not in the processing. It is a defects that is seen during the manufacturing of the can. So when you procure the can for canning, we have to examine all these things. That is why I'm showing you. Okay, the cans they will be sending, but if you find such small places where there is no tin, means it will start spoiling from there. Is it clear? So there will be reactions will be taking place. So if you are in the quality control section, or if you are the in charge of this canning, you have to examine each and every can. So you have to train the staff who is involved there, that they have to examine each and every can before they are going for the canning. Is it clear? So dirt, grease, or other foreign materials on the metal plate prior to coating that can take place. Okay, misapplication of the coating to the metal plates. If you're not coating it properly, there's another problem that we can come across. So these are all defects that we can see in the manufacturing levels of the cans. So pin holes, sometimes we can come across the pin holes, small holes. Okay, this is small holes you can see here, small holes you can see here. Through this hole, sometime the rusting will start. Okay, so these are called as pin hole defects. We can see pin holes defects we can come across. Again, it is a manufacturing defects in the cans. We can, so like that there are many, but these are common defects you can see in the cans. So we have to examine the cans. So you may think this is a very small hole, what is going to happen? No, from here only it will start. Is it clear? From here only the defects in the cans will start. So even in the rust it will start, the rusting will start from here. So paneling, this is usually seen in the what you call as high cans. Okay, when the height of the can is very high, and if you give more edge space than the normal, that's what I told, the edge space should be appropriate. It is 0.6 to 0.9 centimeter you have to give. If you are giving too much of vacuum, too much of head space, means the excessive closure vacuum will be there. So you know how the vacuum is created, because the steam will be there. Once you seal it out, the steam will cool and it will create a vacuum. Is it clear? So the excessive external pressure. So when the external pressure is more, what happens is it will give a paneling like this. Okay. The normal canning in a straight aerium, it will be straight like, but here what we can see, there will be a bending will be taking place, because there is an excessive edge space here. So vacuum is more. Okay. So this is called as paneling. This is called as paneling. So you can see a paneling in large cans. The body of the can is pushed inwards due to high vacuum inside. In severe cases, the seam leakage may occur. So in severe cases, the seam, you know what is seaming here? The joining of two end of the cans, it may leak also. So this is called as paneling. So next is breathers. Breathers means a can with minute leak. This is mainly seen in the seamings. Okay. So there will be minute leak that will permit the air to move inside, and it can cause the spoilage, rancidity and other spoilage can, but it will not allow the microbes to enter. So it will be a very minute holes in the seam region. So in the can seam region, you can see as minute holes which allow the oxygen. What is our basic objective? It should be anaerobic conditions. Here what is happening? The oxygen will start entering, or the atmosphere air will start entering. So the oxidations will be taking place. So that is type of thing is called as a breathers. Breathers means minute holes will be there in the seams. This is mainly seen in the seaming areas. So coming to the spoilage, stages of spoilage, first is the hydrogen swell. So what is hydrogen swell? Here the H2 gas is produced. So when this is mainly not seen in the meat products or the animal products, we will not see. This is most commonly seen in the high acid foods. We will see mainly in the high acid foods. Here you can see the hydrogen gas is produced by when the acids, for example, you say citric acids, lemon you are taking, and you are doing the canning. What happens is this acids can go and react with the metal if the coating is not proper, if the tin coating is not proper. So it will react with the iron, because this is steel, it's made out of steel, so it reacts with the iron. So it forms what you call as the metal salts plus hydrogen. So there will be a release of hydrogen gas. You know that when acid reacts with the metal, it forms metal salts plus hydrogen. So here the case is free of microorganisms and fit for consumption, but we can find a small bulging there. Okay, so hydrogen gas will be released. So the instead of concave, it will form slight convex. Okay, but when we open that such type of cans, we will not have any off odor, or the microorganisms will not seen. And this is mainly seen in the high acid foods. Okay, where the acidity is very high, in such type of food we will come across this type of spoilage, that is hydrogen swell. So if the can is concave, just see that if it is an high acid foods, you can expect it to be a hydrogen swell. So you use your logic and see that. Okay, next is a flipper. Flippers means the can will appear normal. The cans appears normal. When stuck, it will bulge out or flips out, but cannot be pushed back to the normal. So when you just hit against the table or something, it will just flips out. So it is called as a flipper. Then what is springer? A mild swell at one or both end. So one end or the both end, it will be swelling. This is due to insufficient exhaustions or overfilling of the cans, or bulged and pressed back to the original. So what happens here is, the springer means there will be concave, it will be convex, it will be, but once you push it out, it goes to the normal. But after sometimes, just like a spring, it again springs out. Okay, again it's bulged out. So that is called as a springer. So I hope you understood what is flipper. Flipper normal airicum. When you hit it out, what happens? It flips out. Okay, in the springer, it is slight convex, it will be there. When you push it, it will go to the normal. But after some time, again it flips out or springs out. Okay, that is called as springer. Then what is soft swell? Soft swell means, so these are stages it goes like. Okay, flipper, springer, it's a stage by stage it is moving. Okay, soft swell means this is a little more advanced stage. What happens is, there will be swelling at both ends. So when you press it out, it will go to the normal condition. But immediately when you take the pressure, or when you take the fingers out, immediately again it bulges. So that is called as a soft swell. Okay, that is called soft swell. The springer means after some time it swells, but in the soft swells, immediately when you take your hands, it will swells. Then what is hard swell? Hard swell means the bulged end cans cannot be pressed back to the normal positions. So the bulging will be so strong, even when you apply the pressure. Okay, once I tried with our students, I took the hard swell cans and gave to the students. Okay, I asked 3% to push it, but they were not able to do it. So high pressure will be developed, and after that stage, what happens? It will burst out, and the same can burst out in our department. Okay, I kept in a locker, and one day we heard a loud sound, and when we went, we found that it was bursted. Is it clear? So for demonstration, so for students demonstration, to show the students, one day I kept that can, but it bursted out. Is it clear? So that is called as hard swell. That is the final stage. So after that hard swell, and it will burst it out. So the final stage, and the cans will immediately burst. So one day it will burst. So such cans, you take it and dispose it off. Even in the lab, don't keep it. Is it clear? It can burst at any time. Okay, so high pressure will be developed inside the cans. So coming to we have seen different stages of swelling. Now we are going to see the microbial spoilage. First is the flat sour spoilage. Here flat, as the name indicates, it's a flat sour cans. We can see the containers are not swollen. The pH is significantly lowered. Why it is called as a flat sour means the cans will be in the normal concave only, and here the pH will be reduced by, it will give a very sour taste. Okay, this occurs due to slow cooling. If you are not doing the processing, this microbial spoilage, everything is because of under processing. Is it clear? It is because of under processing. So, and if you are not doing the cooling process immediately, I told it should be cooled to less than 39 to 36 degrees centigrade. And if you are not cooling it properly, what happens is the thermophilic species or the facultative anaerobes, that is Bacillus coagulans and Bacillus stereothermophilus. So remember that, Bacillus coagulans and Bacillus stereothermophilus. Here the spores will start growing. Okay, the spores grow at the thermophilic temperature, because you are not doing the cooling immediately. What happens? It's more than 40 degrees centigrade. So at that temperature, it will be more favorable for the spores to germinate, and they will multiply. Vegetative cells will be growing, and the spores will also be growing, will be germinating and produce the acid from the carbohydrate source of the food. So it will produce the acids. It will be acting on the carbohydrate foods, and it will produce the sourness, or it will give the fermented products. So no gas is produced here. So here this is called as a flat sour. Only the sourness of the product will be increasing. It is caused by mainly by the Bacillus coagulans and Bacillus stereothermophilus. Here, because of under processing, and if you're not cooling it properly, the vegetative spores, the vegetative cells plus the spores will also germinate, and it will multiply, and it will act on the carbohydrate source of the food materials, and it gives the fermentation like products it will be forming, and it gives a sour taste. So this is next spoilage, that is called as thermophilic anaerobic spoilage, that is also called as TA spoilage. This is indicated by the swelling due to the production of gases. So this, you know that Clostridium, okay, Clostridium thermosaccharolyticum the bacteria, Clostridium thermosaccharolyticum, it produces the gases. So this bacteria will produce the gases. They produce the gases like carbon dioxide and hydrogen, and it gives a cheesy odor for that product. It gives a cheesy odor. This is again because of under processing, and if your cooling is not proper, what happens is this will germinate. The Clostridium will germinate, and it can produce this type of spoilage. So it develops in the can stored in a hot conditions. Okay, if you are not cooling it properly, if you are not storing it properly, and in that conditions, and if it is under processed, the product is not processed properly, in that cases, this bacteria can survive, or its spores can survive, and it can produce the gases like carbon dioxide and hydrogen, and it gives a cheesy odor. This is one common spoilage we can come across in the animal products. TA spoilage, thermophilic anaerobic spoilage. Next is a sulfide stinger spoilage. Okay, sulfide stinger spoilage. This is caused by Clostridium nigrificens. Okay, Clostridium nigrificens. Again, you can come across in low acid foods. So you know that animal products are low acid foods. So here the hydrogen H2S will be produced. Hydrogen sulfide gas will be produced. You know that this gas is water soluble. So there will not be any bulging. The cans will not bulge. But once you open out, you know the peculiarity of hydrogen sulfide gas. It gives a rotten egg odor. Okay, and you know that it gives a black color. Okay, it gives a black color. So hydrogen sulfide gas will be produced by this type of bacteria, Clostridium nigrificens, and it gives a rotten egg odor, and it will be dark in color. So this is again due to the under processing, not storing at a proper temperature. We are storing at a very high temperature, and no proper cooling is done. So in that conditions, this bacteria will germinate, or the spores will germinate, and they can give to a condition called sulfide stinger spoilage. So once you open it, immediately you get what I call as a rotten egg odor. So I had an experience when I worked with MPI, such type of spoilage can also take place. So it is because of under processing, not heat resistance. Okay, it's not because of not heat resistant. The presence indicator again the under processing. So this is shows that this, because this bacteria will be died normally, because when you are subject to proper time temperature. But here you can see it is not dying. So what happens is this, or indirectly it indicates that it is because of under processing. What is sulfiding? Sulfiding means, see, this is not because of the bacterial spoilage. Again, this problem also we come across. And when you open out, just like sulfide stinger spoilage, you find there is somewhat color change. Okay, the brownish color or blackish color you can come across on the food surface, but you don't get rotten egg odor. It is why? It is because, not because of the Clostridium nigrificens this is caused. It is because why? What happens is in this is that the sulfur will be, you know that meat is a sulfur, it will contain a lot of sulfur containing amino acids. Okay, what happens is this sulfur will be released, or the breakdowns of the protein take place, and this sulfur will be released, and it forms hydrogen sulfide gas. Okay, it forms hydrogen sulfide gas. This hydrogen sulfide gas, what happens is, it is being soluble, it goes and reacts with the iron. If the tin coating is not proper, or we give what a coating called as SR coating, sulfur resistance coating. To prevent the sulfiding only, we give the SR coating, sulfur resistance coating. Okay, if the sulfur resistance coating is not proper, if the tin coating is not proper, then what happens is this hydrogen sulfide will react with the iron, and it forms ferrous sulfide. It forms ferrous sulfide. You know the color of the ferrous sulfide. It will be somewhat brownish black in color. But what is the difference with sulfide stinger spoilage and sulfiding is that there is no rotten egg odor. If rotten egg odor means it is because of the spoilage, that because of Clostridium nigrificens. Here, what is the thing is that it is edible, but appeal or the sensory appeals will not be good. That's the problem with sulfiding. So the reaction continues during storage, and that is the so staining keeps on increasing. This color changes will be keep on increasing. if you are keeping for a very long duration of storage. Okay, so this is called as a sulfiding. So to prevent sulfiding only, we give what we call as SR coating. When I talked in the about the cans, I told the third coating that we give is the second coating that we give is the SR coating, sulfur resistance coating. So what is the sulfur resistance coating? Is that material is that using some phenolic metal lacquer or alkali bath containing phosphates and chromates. Now the latest technology, they also giving some plastic coating is given. Okay, some plastic coating is given, so to prevent the sulfiding. So SR coating is given. Now you understood what's the importance of SR coating or sulfur resistance coating to prevent sulfiding. So some terminologies, again you have to know. One is called as the thermal dead time. The time necessary to kill given number of organisms at a specific temperature. So about this also will be talked in detail in the next coming class on retorting. So what is, just for your introduction, I'm just giving thermal dead time means, see, we give a specific bacteria and we'll be inoculating into the food materials or to the media, and for specific temperature, we keep it at a specific temperature, and we see that how many bacteria will be growing. You understood? We'll be inoculating, suppose 100 bacteria, 100 colonies will be inoculating. Known colonies will get it from labs. Okay, so that known quantity will be inoculated into this media, and we'll subject to a particular temperature. We keep in the water bath, say 121 or 115. Then how many minutes you have to keep? Okay, that is called as thermal dead time. Just I'm giving a rough idea. It is known as F0 value. Okay, D value means decimal reduction value. That is from 100 to 90% reduction. Then suppose there is 100 bacteria. To bring to 10 bacteria, how much time is required at a particular temperature? That is called as decimal reduction value. You able to understand? So what is Perigov factor? Normally in the competitive exam they will ask you in the ICR exams and other things. See, when you use the nitrate along with high temperature, is it clear? So along with the temperature of say around 100, and if you use the nitrate around 120 ppm. Okay, so what is the thing is that it forms a factor called as Perigov factor. It was found by a scientist. It's called as Perigov factor, which has inhibitory power almost 10 times than using nitrate alone. So if you use nitrate alone, suppose it can destroy only one Clostridium means. If you use nitrite along with that, if you subject to 100 degrees centigrade, minimum 100 degrees centigrade, what happens is it can destroy 10 times. So 1 to 10 bacteria you can destroy. You understood that? So heating of meat and meat products with nitrate produce an agent which possesses 10 times more inhibitory power against Clostridium botulinum than using nitrate alone. You got it? If you use nitrate, we have seen that ferredoxin enzymes and all these things we have seen already. But nitrate plus you are heating to high temperature means it will produce a factor called as Perigov factor, which has 10 times more inhibitory power than using nitrate alone. So F0 value for Clostridium, again little objectives I just put for your exam point of view. F0 value for Clostridium botulinum is 2.78 minutes. Okay, 2.78 minutes at 121 degrees centigrade. So normally we take it as 3 minutes. Okay, if the multiple choice they give 2.78 or is the correct one, or in some books we find even 2.45. Okay, so 2.78 normally we take it as 3 minutes. Okay, the bacterial causes liquefaction of jelly of canned ham is caused by Streptococcus liquefaciens. The bacteria cause liquefaction of jelly of canned ham. So you can just note it down. So pre-processed spoilage canned meats. That is, pre-process means before processing. That spoilage is mainly caused by Streptococcus faecalis. Before processing, pre-processing spoilage. I told that sometimes there is a gas. There's a gap between sealing and then processing, retorting. Okay, if there's more gases, sometimes if there is a contaminations, then like Streptococcus faecalis. Okay, so such types of bacteria, if it is present, can be normally present if their personal hygiene is not proper, or the surrounding hygiene is not proper. In that cases, the pre-processed spoilage of the canned meat can take place. This is called this is caused by the Streptococcus faecalis. Another is swelling of canned ham. It is caused by Proteus, swelling of canned hams. So these are certain objective questions. Apart from what I told you, these are certain objective questions you can expect. swelling of canned ham is caused by Proteus species. What is stock burning? Stock burning means if you are overcooking or lack of sufficient cooling after retorting. So if you are not cooling it properly, what happens is we are going to expose to high temperature, or you are going to keep in that high temperature for long durations. So what will happen? It will burn the products. So that is called as stock burning. Number of cans for testing. So normally when we do canning, we have to report to FSSI. Okay, remember, suppose you are working in a canning industry means, or retorting industry means, whenever you do retorting, we have to report to FSSI. That is the rules. Okay, and they will come and take the samples. So how many number of cans or retorts they will take is that up to 500 cans they will take 6 numbers randomly. They will select 6 numbers. 500 to 1000 7 numbers, 1001 to 5008 numbers, 5000 to 10,000 9 numbers, about 10,000 they will take 10 cans. Okay, they can take 10 cans, and they will do the sterility test. And whenever you do canning or retorting, we have to do the sterility test. Then only you have to leave the cans to the market. Okay, so sterility test means we keep it at 55 degrees centigrade. So in the incubator, we keep it, we will say 55 degrees centigrade, and keep around, say how many cans you are doing, approximately that much cans will be taken, and it will kept for 5 to 7 days or even 10 days we will keep it. Okay, 55 degree. So why we keep it at 55 degrees? If you are under process, and since we are keeping at 55 degrees centigrade, the thermophilic spores will germinate. Yes or no? It will be very favorable. This condition will be very favorable for the microbes, thermophilic bacteria to grow. So if your process is not proper, under processed, so in that cases, you will keep it for 5 to 7 days or even 10 days. You will keep it like normally 7 days is enough. 1 week will keep it. And if you see any bulging, or then it is clearly shows that your whole batch is not processed properly. Is it clear? And then that cases, if more cans are showing bulging means, so immediately after canning you cannot send the cans to the market. You remember it. Is it clear? Because if the process is not processed, if the process is under processing, the product is under processed. So at the same time we also keep it at 37 to 35 degrees centigrade also for 10 days. So if mesophilic bacteria is there, that will also be growing. Okay. So again we can see, even the if you are not able to destroy the mesophilic bacteria, again it is under processing. Okay, so like that, different temperature will keep it. Even when I worked with the canning industries, we used to keep it at this temperature. After 5 days or 10 days only we will give, if there is no problem with the cans, then only we will leave that whole lot into the market. Immediately you cannot do that one. But now the technology has developed a lot. The error will be very, very less nowadays. So anyway, we have to see this quality control. The sterility test of the can is very important. So thank you very much. We'll stop here. Take care. God bless you. Okay, bye.
Thank you, sir.